The idea that the gut might influence the brain is no longer a fringe hypothesis. It is one of the most exciting frontiers in modern biology. And now, a new twin study, one of the most rigorous designs we have for teasing apart genetics and environment, suggests that nurturing a healthier microbiome may play a gentle but meaningful role in reducing Alzheimer's risk. The findings don't claim certainty, but they do offer hope, showing early biomarker shifts that point toward possible protection years before cognitive symptoms appear. Twins provide a uniquely powerful window into the gut-brain axis. Identical twins share nearly all their genes and much of their early life environment. So when one twin shows a different metabolic or neurological profile later in life, researchers can look more directly at lifestyle, diet, and microbiome factors rather than attributing differences to genetics alone. In this study, researchers followed older twin pairs who had measurable, though very early, markers associated with Alzheimer's disease, subtle changes in inflammation, minor shifts in cognitive testing, and early abnormal levels of proteins like amyloid or tau. The magic of the study lies in what happened next. The twin with the more diverse and stable microbiome, richer in fiber-loving bacteria, short-chain fatty acid producers, and anti-inflammatory species, tended to show healthier biomarker profiles. Their inflammation markers were lower. Their metabolic signals suggested better insulin sensitivity and mitochondrial function. And their cerebrospinal fluid biomarkers, often the earliest indicators of dementia risk, looked more stable over time. Meanwhile, the co-twin with a less diverse microbiome was more likely to show gradual drift toward patterns associated with neurodegeneration. Because the genetic baseline was identical, the differences pointed cleanly toward the gut. The researchers then expanded the work by analyzing how specific microbial metabolites interacted with the brain. They found that the presence of certain short-chain fatty acids, especially butyrate, correlated with stronger blood-brain barrier integrity. This matters enormously. A leaky, inflamed blood-brain barrier is one of the earliest problems in Alzheimer's development. If the gut produces compounds that help maintain that barrier, it could slow the cascade of changes that eventually lead to cognitive decline. What's even more encouraging is that the study wasn't limited to observation. In a subset of participants, researchers introduced gentle microbiome supportive interventions, higher dietary fiber, targeted probiotics, and increased intake of polyphenol-rich foods like berries and leafy greens. These weren't extreme diets or complicated regimens, just simple, sustainable shifts. Over months, the twin whose microbiome was nudged in a healthier direction showed small but measurable improvements in inflammatory biomarkers and brain-derived neurotrophic factor, BDNF, a molecule tied to learning and memory. The co-twin without the intervention showed no such improvement. While the cognitive measures did not shift dramatically, unsurprising in such a short time span, the biological signals moved in the right direction. It's important to emphasize how groundbreaking this is. Alzheimer's is a disease that begins decades before symptoms appear. If microbiome changes can shift biomarkers earlier in life, that means we may eventually have lifestyle-based tools to delay or soften its trajectory long before medication would even be prescribed. And because the gut is modifiable through diet, sleep, stress management, and occasionally supplementation, the barrier to action is low. This is where the positive potential lies. Intervention is accessible to nearly everyone. Even the study's cautious language carries optimism. The authors didn't declare that microbiome improvement prevents Alzheimer's. Instead, they said it appears to reduce risk by stabilizing biological processes that keep the brain resilient, reduced chronic inflammation, improved glucose metabolism, stronger neural repair systems, and less accumulation of harmful proteins. These are exactly the factors global dementia researchers have been trying to influence for years. This twin study also opens doors for the future. Clinical trials might test whether microbiome-targeted diets can amplify the effects of early Alzheimer's medications. Researchers may identify specific beneficial strains of bacteria that protect neural tissue. Personalized microbiome assessments could become part of routine screenings for people with family histories of dementia. And communities may embrace more fiber-rich, plant-centered eating patterns, not just for heart health, but for clearer thinking and longer-lasting cognitive vitality. Above all, the message here is hopeful. Alzheimer's is complex and multifaceted, but the gut-brain axis gives us a biological lever we can actually influence. Even small habits, daily fiber intake, fermented foods, more vegetables, quality sleep, stress reduction, appear to shift the microbiome in directions associated with healthier aging. The twin design of this study strengthens the case that these changes truly matter. We are still early in understanding exactly how gut ecosystems shape brain destiny, but the direction is promising. This research reminds us that the brain is not isolated. It listens constantly to what happens in the gut. When the gut is balanced, nourished, and functioning well, the brain appears to benefit, not dramatically overnight, but quietly, steadily, over years. And in the quest to protect memory and identity, that steady, accessible, everyday influence may be one of the most powerful tools we have.